Hi, I'm Gina O'Reilly.
I'm Peyton Bailey.
And for our enzyme project, we did aspartate aminotransferase, which we abbreviate as AST throughout this presentation. Before we get started, just an overview of the content we will be discussing. First, we will discuss the structure of AST before getting into the mechanism by which it is synthesized. Then we will compare AST to its sister enzyme ALT before discussing our primary literature, which is focused on alcoholic liver disease. And finally, we will discuss the bioinformatic methods used within the primary literature to assess the AST serum levels.
To begin, we're going to start with the basic structure of our enzyme. Its secondary structure consists of 16 alpha helices and one beta-pleated sheet that has 7 parallel strands and 1 anti-parallel strand. To move on to its tertiary structure, it's a homodimer that each has 412 amino acids in its length. It also exists in 2 isozymal forms, one in the mitochondria and one in the cytosol. The form that we're going to be focusing on today is the cytosol form.
For the domains of this enzyme, there are 2 identical 412 residue subunits, each of which has 2 domains, one small and one large. On the large domain is pyridoxal phosphate, abbreviated as PLP, which is present as a cofactor, meaning its presence is necessary for the enzyme to be able to perform catalysis. On the small domain is the flexible region that changes from open to close as the substrate binds to the enzyme.
The enzyme is encoded by the GOT1 gene that is located on chromosome 14 and includes 9 exons. So, as mentioned before in its name, AST is classified as a transferase enzyme, which means it catalyzes the transfer of a functional group between amino acids. As Gina had mentioned before, it is PLP dependent for the proper transfer of those amino acids. It undergoes 2 similar half reactions, which is also known as enzymatic transamination. And this is the transfer of the alpha amino nitrogen of the aspartate amino acid to an alpha keto acid with the synthesis of a second amino acid as well as a second alpha keto acid. And this is the structure of PLP, as you can see the phosphate group over there. This is the enzyme mechanism that I will be talking about. So initially, as we mentioned before, it begins with PLP. The aspartate enzyme is not shown here, but you can see the lysine 258 amino acid. This lysine interacts with the aldehyde of the PLP, resulting in the formation of a shift bond. This structure then undergoes several more reactions, resulting in the formation of an internal aldimine, an external aldimine, a quinonoid intermediate, as well as a ketamine intermediate, to result in the 2 products, PMP and oxaloacetate. Alpha ketoglutarate will then react with the PMP in the reverse reaction, resulting in the formation again of PLP, as well as a new amino acid, L-glutarate. Oxaloacetate and alpha ketoglutarate play critical roles in the Krebs cycle, so the production of them in this mechanism is very important.
To touch a little more in detail about the mechanism Peyton described, it is a ping-pong mechanism, which you saw in the way that it had a second enzyme complex, different from the first. So in the first half, as Peyton described, an amino acid will react with the enzyme PLP complex and generate an oxaloacetate, in addition to resulting in a modified enzyme PMP complex. Because this is a ping-pong mechanism, we know that the enzyme PMP complex needs to be reverted back to the enzyme PLP complex. And this is done, as Peyton said, when the enzyme PMP complex reacts with the alpha ketoglutarate to produce a second amino acid, regenerating the enzyme PLP complex.
Now we're going to be comparing AST to its sister enzyme ALT. Aspartate transaminase is another name for aspartate aminotransferase. As mentioned before, the reactant amino acid in AST's mechanism is aspartate. Consistently with its sister enzyme, instead of its reactant amino acid being aspartate, it is actually alanine. Both of these are present in many tissues in the body. However, AST's most concentrations can be found in the heart and liver muscles, heart muscles and liver, and the tissue concentration for ALT is in the liver and the kidneys. This is important that these top two concentrations are found in the liver, as our primary literature focuses on the liver serum levels of these 2 enzymes. Both of these use PLP as a cofactor, and both of these are classified as transaminases, meaning that they will both undergo a similar mechanism that I had mentioned earlier. Both of these hold clinical significance in determining the detection and differential between many diseases of the liver. The reason that these can be measured in blood serum levels is due to membrane blebbing. Membrane blebbing is when smooth circular extensions of the plasma membrane expand and are eventually released into the bloodstream. Irreversible damage occurs when these membrane forms bleb off and the plasma membrane does not repair where that blebosome came off, resulting in a hole in the plasma membrane, which allows the enzymes that are in the cytosol, such as AST and ALT, to be released into the bloodstream. Reversible damage, however, occurs when the blebosome comes off, but the plasma membrane is able to repair where it was released from. Again, this is why we are able to measure the level of these enzymes in the blood levels. You can use AST and ALT in a ratio to determine the extent of the liver damage that you're focusing on. The higher the ratio indicates things such as alcoholic liver disease, and a lower ratio indicates things as viral hepatitis. We're going to be focusing on alcoholic liver disease today.
As Peyton noted, AST is not only found in the liver but also in the heart and many other organs throughout the body. This is important to note before we get into our literature because from a clinical standpoint, while AST and ALT levels in a serum blood test can show the etiological liver disease, it cannot predict the source from which the AST or ALT was derived. This is important when we think in the context of someone who may have dermatomyositis, which is an inflammatory disease of the skin and muscles. This disease also results in elevated AST levels. This is why in our literature they use multiple bioinformatic methods to verify that the increased AST levels is from liver necrosis.
So as I mentioned before, our primary literature focuses on alcoholic liver disease. Alcoholic liver disease is when the liver is chronically damaged from alcoholic intake. Symptoms of this disease include dry mouth and increased thirst, abdominal pain and swelling, yellowing of your skin and your eyes, as well as weight loss. This leads to a chronic elevation in AST levels, and depending on the acuity or the severity of someone's condition, can cause a fluctuation in these levels. The end stage of this disease is cirrhosis, which is irreversible, and usually people with cirrhosis, as mentioned before, will release more AST into their bloodstream. The name of our primary literature is Transcriptome Analysis of Garlic-Induced Hepatoprotection Against Alcoholic Fatty Liver. The goal of this experiment was to test the molecular mechanisms of garlic oil, abbreviated as GO, and diallyl sulfide, abbreviated as DADS, or DADS, to test their molecular mechanisms on hepatoprotection against alcoholic-induced fatty liver. For their methods, they took 18 experimental mice and fed them an ethanol diet for 18 days to induce an alcoholic fatty liver. These 18 mice were then divided into groups of 6, each receiving a different treatment. 6 of them received the garlic oil treatment, 6 of them received the diallyl sulfide treatment, and 1 group just received the alcoholic diet and no treatment to induce hepatoprotection. These mice were then sacrificed on day 29 and their blood was collected via a cardiac puncture. From this blood that was collected, the AST levels were measured.
In addition to the method Peyton just discussed, they also performed a microarray analysis to verify whatever biochemical results they obtained from the serum. They did this by taking the liver tissue, grinding it up, and extracting the DNA, from which they transcribed the cRNA single strand. This strand was able to be processed through the Ingenuity pathway analysis software and produce a p-value. In the set of these circumstances, the p-value assesses the likelihood of gene input on the given pathway, like Peyton said, to see the molecular level of which hepatoprotection was productive. The transcripts were then processed through the Panther bioinformatic program to assess the molecular function, biological processes, and the Panther class.
So these are the results from our primary literature, and we're going to be focusing on this mini graph up here in this corner. As you can see, on the y-axis we have AST blood serum levels, and down here are the different types of treatment that each mice received. This first column is the control group of mice that did not receive any ethanol diet or any hepatoprotection treatment. The second group is the mice that received the alcohol diet but no hepatic treatment. The third group is the ethanol mice who received the DADS treatment, and the fourth is the GO treatment for the mice with the ethanol diet. As you can see, the alcohol diet without any hepatoprotection mechanism had the highest level of AST in their blood serum. DADS, which is the first group that received, which is a group that received a hepatoprotection treatment, shows a decrease from the alcoholic negative control group, however, not very significant. The garlic oil group, however, showed a significant decrease that to almost the normal levels that the control group mice had. This is important because it shows that AST does in fact serve as a reliable marker for liver damage. And this is because the chronic alcohol abuse for these mice over the period of 18 days resulted in liver damage along with inflammation of the liver cells. And inflammation of these liver cells led to increased impermeability of the liver cell membrane, resulting in the leaking and membrane blebbing of the AST and ALT enzymes into the bloodstream.
The ultimate goal of the microarray analysis was to use the results generated from the Panther bioinformatic program to see an enrichment analysis of the transcripts that were produced from that process. However, before they could move forward with that, they performed a qRT-PCR to make sure that the microarray data was valid. And they did this by selecting genes and normalizing them to the control GAPDH. And all 5 of these genes, as you can see in the image here, showed consistent patterns with the microarray data, demonstrated by in all of these graphs. You can see how the darker shaded bar, representing the qRT-PCR, and the lighter shaded bar, representing the microarray results, are all trending in the same way, which verifies the microarray data and allowed the experimenters to move forward with the enrichment analysis. For the enrichment analysis, 43 transcribes were found to be initially perturbed, but then restored by the DADS and/or GO treatments. To break this down in terms of the methods used, 36 were restored by DADS, 17 were restored by GO, and 10 were restored by both. The top 2 gene networks that were developed using the Panther program can be seen on the next slide. So as you can see here, these are 2 separate networks for each of the treatment groups, this one being the DADS treatment and this one being the GO treatment group. There are a variety of red and yellow indicators for each gene, the red being genes that were upregulated and the green being genes that were downregulated. Most notably in this set of data is that in this pathway and this pathway, these have to do with lipid metabolism, and in both of the treatment groups, this was seen in an increase. It is also necessary to note that this is the second most altered network for DADS, while here the lipid metabolism network was the most prevalent in the GO treatment, showing, like as Peyton said, that the GO had significantly less AST serum levels than the DADS treatment did. And this is significant because lipid metabolism being upregulated means it's able to combat the amount of lipid droplet that you see with fatty alcoholic liver disease, verifying both the serum results and providing the information needed to make the conclusion that there was hepatoprotection given by these treatments. In conclusion, the goal of the literature was to assess the hepatoprotection, and both of the treatment groups were able to do that, and AST helped to see to what extent each treatment group did that. The bioinformatics methods were then used to verify the AST levels came from the liver and were able to provide the information as to why the AST levels were prevalent in that manner.
As mentioned before, the enzyme AST and its mechanism is very prevalent in things such as the Krebs cycle, which gives the body energy in terms of its reactants and products, oxaloacetate and alpha-ketoglutarate. This is important because not only does AST important for the body, but it also is reliable in terms of determining the disease and etiology on the body.